There are a couple of minor exceptions to these trends, but the overwhelming number of organisms show patterns like this. It's a little bit different in the oceans too, but broadly it holds. So why is it so high in the tropics diversity? One thing you can think about in the deeper time perspective is glaciation. We've gone through this series of ice ages over the last millions of years, particularly over the last million, these cyclical ice ages, where when it's really cold, you get the glaciers that form to the north and migrate south. And then in the warming portion of the cycle, they retreat. And then when they retreat, they're leaving a scoured landscape, much like you saw in Glacier Bay, Alaska, with the retreat of that glacier. And they're setting the successional sequence back to zero. Having covered this land and wiped out any of the forests, when they retreat, it's back to square one for those systems in a really deep time perspective, not just in the successional context. Whereas the equatorial environments are not under ice during these ice ages. The sea level changes a lot because when you lock up all this water in ice, these sea levels might go down. They do go down. Sea level can change. The forests can become cooler when it's globally cooler. The precipitation regimes can change in the tropics. So it's not like they're uninfluenced by the ice ages, but they're not covered in ice. And that's a big difference. So refuge from glaciation at the low latitudes, refuge for the species that live there. Whereas the species that are typical of these areas will have to migrate to survive, or they'll be wiped out. So that could play a role. Let's look at the structure of tropical systems, in particular the forests. They're really complex. Such forests are mosaics of habitats, partly because of the regularity of disturbances, small disturbances like tree falls. When a tree falls in a tropical forest, it brings down a lot of other material with it. Not only some of these huge trees, particularly the big emergent trees that grow so massively in these forests, they not only knock down other trees when they fall, but they may be strung together with lianas and vines that they pull a bunch of other trees with it, and they create these massive gaps. That's going to happen in all forests, but tropical forests with their thin soils are particularly good examples of these types of disturbance, and the regularity of such disturbances can help to increase diversity. And if you think about it in the intermediate disturbance hypothesis context. Somewhat related is that structural complexity of a tropical forest. Three-dimensional structural complexity has a big influence on the organisms that are associated with it. If this were just a level and flat ground, it's very different from the hyper-complex environment with all these physical and spatial little crevices and niches, niches in a spatial sense, niches in an ecological sense, that are available in a very complex system like this. These systems are structurally complex. They have those big emergent trees that tropical forests will, a full canopy that's closed. You have the climbing, the vines and lianas, and then you have the understory plants, and then you have the whole herb layer and the ground layer, and then not to mention the whole subterranean biota. They're very complex systems. If you think about what a forest looks like up in the Sierra Nevadas, if you go up it, it's not nearly as complex spatially, structurally, as a system like that. That may play a role. Certainly a role is played by the abiotic factors of temperature and moisture. There's a whole lot of water in these warm systems, and that influences the pace of life, on deep time scales and shallow time scales in ways that are interesting and that you can think about. The evidence is like this. When you map something like tree species richness in the Americas relative to evapotranspiration, which is a variable that really combines within it temperature and moisture. You haven't had your botany, so I'm not going to unpack that too much, but let me just tell you that it involves temperature and moisture, how much water evaporates from the land surface and how much transpires from the plant leaves. And that's related to how much water is in the system and how warm it is. So the warmth and wetness of the system relates to the diversity or the richness in a positive correlation like that. I was looking at this paper and thought it was interesting because this shows sort of a never diminishing increase in richness relative to evapotranspiration. But if you get to really high levels of evapotranspiration where it's really warm and really wet, maybe you see a decline in richness related to that. This graph just stops before those levels. So it's interesting to think about too warm and too wet and what that means for richness. Vertebrates. So vertebrates that don't fly. Look at them in relation to evapotranspiration. Potential versus actual, don't worry about it. We can talk about it in office hours if you want. It's not important. Same idea, same relationship. So to summarize some of those points, you can write this down later from your PDF, but I'm summarizing some of that for you. The relationship between ambient energy and moisture, ambient energy like solar input, that's hot and wet in the tropics. There's stability, long periods of stability without cataclysmic disturbances. Think about your graph of disturbance intensities and scales. Well, a glacier covering the landscape is an intense, spatially extensive type of disturbance. The complexity of tropical systems in a structural and three-dimensional sense. Could the interspecific interactions play a role in generating or maintaining diversity in the tropics? All the predation and mutualism and highly competitive relationships in those systems, can diversity and richness kind of build on itself in the context of complex interspecific interactions in a spatially complex three-dimensional environment over long periods of time with a lot of moisture and warm temperatures? These things are not mutually exclusive in explaining tropical diversity. It could be a multifactorial explanation. The Galapagos are a bunch of islands, volcanically produced, that have different shapes and sizes. The mainland is over here. South America is in this direction, to the east. And these islands are interesting because of their differences in relation to their biota, among many other reasons why they're interesting. If you think about the species area curves that we saw, well, they should apply in this island context, right? These are just land masses of different sizes. Wouldn't you expect the species area curve, number of species, to hold on a log-log scale? Shouldn't it be aligned? Yeah, in theory, it should. And for something like the plants of the Galapagos, it tends to work. The larger ones have more plants consistent with a linear relationship on a log-log graph in an island context. And remember that these are islands in the classic definition of an island. But this could be a lake in a sea of land, right? This could be an aquatic environment in a terrestrial environment rather than a terrestrial environment in an aquatic environment. And it would still be an insular ecosystem. Or it could be a mountaintop in a region of lowland. And these would be mountaintops in a low continental system. They would be insular in ecosystems, mountaintops, in a very different habitat. So don't restrict your notion of islands to the classic type. It's generalizable. Parks in an urban environment. These could be parks in Los Angeles, something like that. Those would be insular environments in an urban environment. The West Indies, the Caribbean, islands of different size. The herps, the amphibians and reptiles of those islands in relation to the size of the island. Just another example to reinforce the empirical reality of those relationships for islands. and an example from ants. New Guinea, the relationship of the species area curve on New Guinea is different from the islands off of New Guinea. And this is where you can get into the, well, why? Why is that line, why is the intercept different for that line? Why is the slope of it different relative to the islands? Those are things we won't get into, but are really interesting about biogeography. A formal theory of island biogeography developed from the collaborative work of E.O. Wilson and Robert MacArthur. MacArthur was the more mathematically inclined. E.O. Wilson was the great naturalist and field biologist, and they got together and they produced a really influential series of papers and an influential book that attempted to put some more mathematical underpinning behind the study of islands in a biogeographic context. And they were interested in how we can explain how many species there are on different islands. And they related it to the rates of colonization of those islands, and based on the distance of the islands from their sources and the sizes of the islands. So let's look at a few of the graphs to help understand that. Number of species, species richness on the x-axis, rates of immigration or extinction on the y-axis. The immigration curve, the number of species arriving on an island, and the extinction rate, how quickly they're lost from an island. Where those curves cross, the authors interpreted that as the equilibrium number, the predicted number of species that would exist on the island. And what we need to see is how those rates of immigration and extinction are related to size and distance of the islands. So a large island is going to have higher immigration rates of new species colonizing it from a mainland or from the source area because it's bigger. It's a bigger target. And when species are propagating, they're more likely to hit the bigger target than they are the small target. And so your immigration rates are going to be higher for the bigger mass. The extinction rates are going to be higher on the smaller mass than they are on the larger mass. What's one reason? Why? Maybe from your evolution section, why you would think that the smaller island would have higher extinction rates? Fewer resources. Yeah? What's something else? Something related? Yes? Smaller gene pools, smaller population sizes, more likely to drift to extinction. One of the reasons. Then on a larger island, where the extinction rates will be lower. So size of island plays a role in determining your equilibrium number of species, according to this theory. Distance of island, the distance effect. Near islands are going to accumulate species by immigration faster than far islands because they're closer. If you shoot out a bunch of species from a mainland, they're going to hit the near islands with greater likelihood than they are going to hit more distant islands, just by chance. So nearer and bigger is going to be colonized more quickly than farther and smaller, farther and smaller. Replacement rates, we'll get into more of it in time. What the authors wanted to do, and this was, I guess that clicks away. What the authors wanted to do, and this was particularly E.O. Wilson, now with one of his students, Daniel Simberloff. MacArthur died at a young age, and Wilson wanted to carry on with some experimental work, and one of his students, a graduate student, was motivated to do this work, to do this testing in the field. They worked off of Florida on these little mangrove islets, these little patches of mangrove that could actually form islands, small islets that could become larger islands. They varied in size, and they varied in distance from the shoreline. But they could be little like this, and just near the shore, they could be larger like this. And they've got cormorants on it, that's an osprey. You can imagine all the insects, more cormorants, birds, different plants even growing epiphytically on these other plants. It's a more diverse community there than that small one we looked at, probably. A distant one that's much larger still. So it's a neat opportunity to study some of this experimentally if you can manipulate the systems. And that's what they did. This was, I'm not sure you could get away with this in this day and age. I'm not sure you could get the permits from the state of Florida to go put a scaffold and a tent over these islets and fumigate them with methyl bromide. It doesn't kill the plants, but it kills all the arthropods, the insects and friends. And they did that experimentally to a series of these little islets and then watched how they were recolonized and followed them over time in terms of the species that occupied them in order to test their theories of island biogeography. And this has influenced a lot of subsequent work in conservation biology and basic ecology. Some of the data for a series of islands, 4 different islands, all about the same size, all about 10 meters long. So size is held constant here, but distance to mainland varies. And what they follow are the number of arthropod species that recolonize those islands based on how far they are from the mainland. And trace it over 2 years. An island like this that's very far from the mainland ends up at the end of the study with this many species. At the start of the study, it had this many species, about 25. At the end of the study, it's approaching 20. Or this island here ends up with about 28 species or something. At the start of the study, it had about 31. The red dot, it's 172 meters from the shore. That one was 533 meters from the mainland. And the one that's really close, just a couple meters of that size, started with a bunch of species, more than 40, and it ended with almost as many. So what the authors interpreted this as was a recovery to equilibrium numbers of the species. And it varied, the rapidity with which they arrived back at their equilibrium varied in relation to distance from mainland. And that after some time, they seemed to look like they were stabilizing in numbers. What another set of investigators did with the same data after the study was to take the same data and reinterpret it. So I want you to think about this, because all these guys are doing, and it's a heck of a lot of work. You have to be able to identify, you have to be a really good naturalist to be able to identify all these species in a sample, even if they're just arthropods. You have to be quite the expert there, not to mention all the work that you have to do in the field. But it can be fun. They were interpreting it in number of species, counting out the species. What another group of investigators came along and did was look at the same data by functional groups, by ecological groupings or trophic groupings. For one thing, they broke it down into, well, of those arthropods, how many were herbivores, how many were carnivores, and how many were parasites? And you could follow that trend just by categorizing those same species. You don't even have to go to the islets to do this work. You can sit in your library at home and take their species list, categorize them into these different functional groups, and reinterpret the data. And when they did that, breaking the organisms down into all these different categories, the detritivores, the scavengers, the herbivores, the ants, it's kind of a strange category because it's a non-functional category. When they did it and they compared the before and after richness of each of the categories, they found constancy in those relationships, before and after, that the ecosystems, these communities, seem to recover their original richness by functional category as well. So they interpreted this as the communities having recovered its ecological structure over time. And you can click it here if you want to read that little note. It was just a tiny little note, stimulated a lot of interesting discussion back then. And I want you to think about the difference between taxonomic richness and functional richness. And I wish you a great weekend.